Hello everyone and welcome to vEgyan Ghar's YouTube channel. Today we shall discuss the 2020 TIFR GS paper. TIFR GS is a nationwide entrance exam conducted by Tata Institute of Fundamental Research for admissions in postgraduate courses offered by it. The paper has 4 sections with 15 questions each. The sections are General Science, Physics, Chemistry and Biology. In this video, we shall discuss the chemistry and biology sections of the paper. Here is the chemistry section. The types of bonds present in NH4Cl are A. Ionic, covalent and coordinate, B. Only ionic, C. Ionic and covalent, or D. Covalent and coordinate. For this, we draw the structure of NH4Cl. When we do this, we understand that the bonds between N and H are covalent bonds, whereas this directional bond between N and H is the coordinate bond, and the bonding between Cl- ion and NH4 plus ion is an ionic bond. So the correct answer is option A. The product formed is an isobar if there is A. 1 alpha emission, B. 1 beta emission, C. 1 alpha and 1 beta emission, or D. 2 alpha and 1 beta emission. Now, when we talk about isobars have the same mass number. Only beta emission does not change the mass number. So the correct answer is option B, 1 beta emission. Which element has the greater difference between the first ionization energy and the second ionization energy among potassium, calcium and gallium? A. Potassium, B. Calcium, C. Gallium, or D. All have the same difference as they are in the same row of the periodic table. In case of potassium, the electronic configuration is as shown here. So once the outermost electron is removed, it would be very difficult to remove the next electron as the remaining configuration is highly stable. So the correct answer is option A, that is potassium. Which of the following can chelate metals among salicylaldehyde, salicylic acid and O-nitrophenol? A. Salicylaldehyde, B. Salicylic acid, C. O-nitrophenol, or D. All of these. When we draw these structures, we understand that all these chemicals have heteroatoms having lone pair of electrons, such as nitrogen and oxygen. So we see oxygen and nitrogen. So since they have lone pair of electrons, they can chelate metal ions. So the correct answer is option D, that is all of these. Two solutions of the same molecule with concentrations of 10 raised to minus 3 molar and 5 into 10 raised to minus 4 molar are kept in two different cuvettes with path lengths 1 cm and 2 cm respectively. If the absorbance of the first sample is 1.2, what will be the absorbance for the second sample? A 1, B 1.2, C 2.4, or D 6.0. Now, in this question, from case 1, we will calculate the molar extinction coefficient and use it to calculate the absorbance in the second case. So to do that, in the first case, 10 raised to minus 3 molar solution is kept at 1 cm path length to give absorbance value of 1.2. When we use the Beer-Lambert's law with the formula A is equal to epsilon dot C dot L, where epsilon is the molar extinction coefficient, C is the concentration, and A is the path length, we get the epsilon value, which is 1200 molar inverse centimeter inverse. Using this epsilon value, we will calculate the absorbance for 5 into 10 raised to minus 4 molar concentration in a cuvette with path length 2 cm. Using the same formula, we get absorbance is equal to 1.2. So the correct answer is option B. In the following structure, the ease of breaking of phosphate bonds follows the order A 1 greater than 2 greater than 3, B 3 greater than 2 greater than 1, C 2 greater than 3 greater than 1, or D, 1 is equal to 2 is equal to 3. Now, in case of this given structure, the removal of gamma phosphate, the last one, is preferred as it eases out the negative charge repulsion. Inorganic phosphate is released and the remaining structure is stabilized by resonance. Also, ADP has a high degree of solvation. So the correct order would be gamma greater than beta greater than alpha, which according to the original numbering is 1 greater than 2 greater than 3. So the correct answer is option A. Among the following salts, which has the highest water solubility? A. NaClO4, B. CaClO4, C. LiClO4, or D. KClO4. Now, water solubility is based on the hydration energy. The bigger the size of the atom, lower is this hydration energy. Out of these, lithium is the smallest atom given in these salts. Lithium perchlorate, that is LiClO4, is best known for its enormous water solubility. This is due to its lowest size. So the correct answer is option C. What is the hybridization of triplet carbene? A. sp3, B. sp2, C. sp2 and sp, or D. sp. Triplet carbene may adopt a bent or linear conformation and accordingly may have sp or sp2 hybridization. This you can understand from here, where it has observed a bent shape in case of sp2 and linear in case of sp. So the correct answer is option C, sp2 and sp. Choose the molecule that has polar bonds but does not orient within an electric field. A. HCl, B. Cl2, C. CCl4, or D. NH3. Out of these, Cl2 is homodiatomic. So, there is no dipole moment or polarity. Therefore, there is no electric field. HCl and NH3 are having dipole moment. In case of CCl4 is tetrahedral symmetrical. Even though it is polar, the net dipole moment is 0 and hence it is non-responsive towards electric field. So the correct answer is option C. A monobasic acid has pKa is equal to 5.2. Calculate the pH at which the ratio between the acid and the anion, that is A-, will be 1:50. A 3.5, B 5.7, C 6.9, or D 7.4. Using the Henderson-Hasselbalch equation, we can find the answer for this question. The equation is as follows. pH is equal to pKa plus log base by acid. After putting in the values, we get pH is equal to 5.2 plus log 50 is by 1. So the pH is equal to 5.2 plus 1.7, that is 6.9. So the correct answer is option C. Arrange the following compounds in increasing order of acid strength. First phenol, second O-nitrophenol, third M-nitrophenol, fourth 2,4 dinitrophenol, fifth 2,4,6 trinitrophenol. The options are in increasing order: 1, 5, 4, 2, 3, B 5, 4, 3, 2, 1, C 1, 3, 2, 4, 5, or D 1, 2, 3, 4 and 5. Now first we shall see the structures of each of these compounds. They are as follows. We should know that the presence of more electron withdrawing groups nearby increases the corresponding acidity or potential to lose a proton. So more electron withdrawing groups give -I or resonance effects, which means more acidity. Out of these, we can see that the most acidic compound is 2,4,6 trinitrophenol, followed by 2,4 dinitrophenol, followed by O-nitrophenol, which is more stable than M-nitrophenol because of the intramolecular hydrogen bonding, followed by M-nitrophenol, and finally phenol. So the correct answer is option C. Between metal carbonyl complexes A, that is MCO4PET3, and B, MCO4PPH3, the ability to act as a catalyst for nucleophilic addition to olefin is A, similar for both, B, higher for A, C, higher for B, or D, indeterminate based on the given information alone. Now, these are metal carbonyl coordination compounds of transition metals with carbon monoxide as the ligand. In case of B, the presence of triphenyl rings makes it better candidate as a catalyst for nucleophilic addition to olefin. So the correct answer is option C, that is higher for B. An acidic solution containing 0.01 molar La3 plus is treated with NaOH until LaOH thrice precipitates. At what pH does this occur? The Ksp given is 2 into 10 raised to minus 21. Option A 6.8, option B 7, C 7.8, or D 8. This can be calculated as follows. LaOH thrice gives 1 La3 plus ion plus 3 OH minus ion. Ksp can be calculated as the concentration of La3 plus into the concentration of OH minus. After putting in the values, we get the concentration of OH minus ion to be 2.63 into 10 raised to minus 7 molar. Since this solution is too dilute, we should consider the impact of water. So the total OH minus concentration will be equal to 2.63 into 10 raised to minus 7 plus 10 raised to minus 7. So the POH will be 6.44, or the pH will be 14 minus 6.44, which is equal to 7.56. So the closest answer would be 7.8. The correct answer is option C. A molecule has formula C4H8O2. Based on the following proton NMR spectrum, what is the structure? Is it A, B, C, or D? If we draw these structures, we can find out a bit more clearly that only A and D can give rise to 3 signal peaks. But in case of D, both the peaks for CH2CH3 should show splitting behavior. Hence, option A is correct. Which of the options listed below show the most appropriate set of reagents to carry out the following reaction? Option A, first ETMGBr followed by HCl. Option B, first HCl followed by ETMGBr. C, first C2H5Li followed by HCl. Or D, first C2H5 twice CuLi followed by HCl. HCl followed by the Grignard reagent will give this product. So the correct answer is option B. Now we move on to the biology section. Ocean acidification has multiple consequences on marine life, including reducing the availability of carbonate for skeletal formation of the ocean organisms, increasing coral bleaching, and other impacts. The main driver of the increasing acidity of our oceans is A. Increased ozone that it reacts with water to release hydrogen atoms. B. Increased plastic pollution which breaks down in the water. C. Increased CO2 that reacts with water to form carbonic acid and then bicarbonate. Or D. Reduction in marine life which decreases the amount of ammonia waste. Release of carbon dioxide due to human activity has changed the pH of oceans from 8.25 to 8.14 in the period between 1751 to 1996. This has increased H plus concentration by up to 30%. So the correct answer is option C. Increase CO2 that reacts with water to form carbonic acid and then bicarbonate. Which one of the following would produce the highest variability of gametes if the alleles were assorted independently? And here are the four options. So when we plan out these four options, we get something like this. Now, in case of heterozygous, all combinations produce more variables. So in case of option B, we can find that the variability is very high. So the correct answer is option B because of the presence of heterozygous alleles. In PCR, annealing temperature of the primer does not depend on A. Primer length, B. DNTP concentration, C. The secondary structure of the primer, or D. The GC content of the primer. In PCR, the annealing temperature depends upon the primer length, GC content, primer structure, and salt concentration. So the correct answer is option B. It does not depend on the DNTP concentration. In a new species of bacteria, tRNA anticodons are recognized by 2 nucleotide bases instead of 3. What would be the number of amino acids and the number of peptide bonds respectively of a polypeptide encoded by a 120 nucleotide long coding DNA sequence, which includes a 2 base stop codon? Now, 120 nucleotides gives 59 codons plus 1 stop codon. Hence, we can find 59 amino acids which will be connected by 59 minus 1, that is 58 peptide bonds. So the correct answer is option B, 59 and 58. Which of the following statements is incorrect about meiosis? A. 2 rounds of DNA replication occur. B. The chromosome number is reduced to half. C. 4 daughter cells are formed. Or D. Homologous chromosomes are paired. In meiosis, only one replication stage occurs at interphase 1. So the correct answer is option A. The recent All India Tiger Census counted all individuals above the age of 1 year as adults. An earlier estimate 4 years ago included only individuals above the age of 1.5 years. Given this information alone, can these two estimates be compared? A. Yes, because the age of the first reproduction in tigers has decreased due to pollution. B. No, because they are comparing different sets of tigers in the population. C. Yes, because these are random samples of the whole population. Or D. No, because many tigers die specifically between 1 and 1.5 years. The two estimates cannot be compared due to different set of tigers in the population. So the correct answer is option B. Dogs are genetically very close to wolves, yet all wolves look similar while the breeds of dogs look very different from one another. This is best explained by which of the following? A. Wolves have very little genetic variation as a species. B. Wolves are selected for camouflage. C. Different dog breeds have been created by artificial selection and inbreeding. Or D. Dogs evolved more recently than wolves. Dog breeding is a prime example of artificial selection, and human interference has drastically altered the appearance of dogs. This is called selective breeding. So the correct answer is option C. A student was asked to clone a DNA fragment flanked by BAM-HI with the restriction site GGATCC sites into a vector cut with BG3 with the restriction site AGATCT. The cloning was successful as these two restriction enzymes are known to create compatible sites. Based on this, what will be the restriction site configurations of these two enzymes, where the slash indicates the point of enzymatic cleavage? A. G/GATCC and AGATC/T, or B, G/GATCC and A/GATCT, C, GGATC/C and A/GATCT, or D, GGATC/C and AGA/TCT. Now, BAM-HI would cut at G/GATCC and BG3 would cut at A/GATCT. Here, both REs must produce similar overhangs, which is only possible in case of option B. The correct answer is option B. A student has a mixture of different proteins. The molecular weight of protein A is 30 kilodalton, protein B is 35 kilodalton, protein C is 40 kilodalton, and protein D is 50 kilodalton. The protein mixture was purified using FPLC size exclusion, that is gel filtration chromatography. What size of fraction will elute first? A. Protein A with weight 30 kilodalton, B. Protein B with 35 kilodalton, C. Protein C with 40 kilodalton, or D. Protein D with 50 kilodalton. From this diagram, we can see that the high molecular weight fraction in gel filtration elutes first in the void volume, while the low molecular weight protein comes later. So the correct answer is option D. A hydrola plant is growing in a beaker with water. What will happen to the rate of photosynthesis if you add sodium bicarbonate to the water? A. It will remain unchanged. B. It will decrease. C. It will stop. Or D. It will increase. Increased sodium bicarbonate will provide more CO2, which increases the photosynthetic rate. So the correct answer is option D. You decide to improve your Western blotting by customizing the buffer used for incubation and washing to hear from your friends that they routinely use reducing agents to purify proteins. So you incorporate DTT into the Western blotting procedures and realize that it only makes it worse. You do not get any signals. The reason for this is A. DTT inhibits horseradish peroxidase conjugated to your secondary antibody. B. DTT prevents the binding of secondary antibodies to primary antibodies. C. DTT specifically blocks the interaction of primary antibody with antigens. Or D. DTT destroys the structural integrity of the antibody. DTT is a reducing agent which may degrade the antibody structure by reducing a lot of disulfide bonds required to maintain the antibody structure. So the correct answer is option D. In the following pedigree, what will be the genotype of the parent indicated by the dark square? Dark symbols indicate individuals with a specific phenotype controlled by the locus with the alleles capital A or small a. A, capital A, B, capital A, C, small a, or D, it could be either capital A, or small a. The given chart shows autosomal dominant trait. Hence, the individual, that is the parent, with the dark square has to be heterozygous, that is capital A, small a. So the correct answer is option B. A bacterial gene has two alleles, A and B. A scientist measured bacteria grown under two different treatments in the laboratory, and she measured proportion of the population that had allele A, repeating the experiment 6 times in each case. The results are shown below. Which of the following statements could you make about the alleles? A. Allele B is beneficial in treatment 1. B. Allele A is beneficial in treatment 2. C. Allele B is beneficial in both treatments. Or D. The fate of allele A is more variable than that of allele B. In the second experiment, the frequency of allele A is more augmenting. Hence, allele A is more beneficial in treatment 2. So, correct answer is option B. A protein coding gene in the mouse was mutated such that the resultant protein was non-functional. The phenotypic effect of the mutation was observed in homozygous mutant mice in heterozygous mice that inherited the mutation from the father. This suggests that A. The gene is imprinted, B. The mutation acts as a dominant negative, C. The gene possibly has more than two alleles, or D. The gene is X-linked. We can understand from this given data that the mutation acts as a dominant negative. So the correct answer is option B. A single-stranded RNA virus was grown in cell culture, either in the absence or in the presence of the chemicals X or Y. The intracellular genome copy number of the virus and the titer in the cell culture supernatant, that is plaque-forming units, were estimated over several hours. Based on the data presented below, choose the most correct option. A. X inhibits viral entry and Y inhibits viral genome replication. B. X inhibits viral genome replication and B inhibits viral entry. C. X inhibits viral release and Y inhibits viral entry. Or D. X inhibits viral entry and Y inhibits viral release. In the given graph, X inhibits viral entry while Y inhibits its release. So the correct answer is option D. Thank you for watching. Please like, share and subscribe to vEgyan Girls YouTube channel for more such videos.